Welcome everybody to today's Penn AI Tech webinar. Penn AI Tech is the Penn Artificial Intelligence and Technology Collaboratory for Healthy Aging, an initiative funded by the National Institute on Aging. Today, we're very happy to have with us Dr. Sil Kar, who is a full-time assistant scientist at the School of Medicine, Department of Biomedical Engineering at Emory University. Her research is mainly focused on characterization of unique imaging features within optical coherence tomography, fluorescent angiography, and retinal fundus images associated with disease prognosis, potential treatment response, and potentially disease identification. She's particularly interested in developing computational imaging biomarkers to predict and monitor anti-VEGF therapy response in patients with diabetic retinopathy and diabetic macular edema, as well as elucidating their biological and molecular underpinnings. In addition, Dr. Sil Kar's work extends to exploring the associations between retinal biomarkers and various neurodegenerative and systemic diseases, contributing to the emerging field of oculomics, the study of the eye as a window to overall health and brain function. So with that, I want to remind our attendees that you can use the Q&A feature on Zoom to post any questions you have for our speaker, and I'll be back at the end of the talk for that actual Q&A session. So with that, I'll turn it over to Dr. Sil Kar. Thank you again for joining us today.
Thanks, George, for the nice introduction, and thanks for giving me the opportunity to present some of the works that we are doing at Emory University, and in collaboration with Emory Brain Health Institute and some other institute in the USA and also abroad. So I am the assistant scientist in Emory University, Madhubushi Lab, School of Medicine, Department of Biomedical Engineering, Emory University. And in our lab, we have been working using artificial intelligence technology for different types of disease diagnosis, prediction of treatment response, and disease prognosis. And I am mainly focused on, my work is mainly focused on different ophthalmology related works where we look at different retinal features from different OCT scans or fundus images. And from those images, we could try to find out the retinal patterns that are associated with different systemic disorders like cardiovascular disease, chronic kidney disease, and different neurodegenerative disorders, including Alzheimer's disease. So today I will be discussing that how we could apply artificial intelligence technologies for diagnosis of AD, but not limited to the diagnosis of AD, but how we could use these artificial intelligence models for more diverse implications. So a little bit of introduction here. Alzheimer's disease and related dementia are the most common cause of neurodegeneration, and they have become global epidemics affecting more than 55 million individuals worldwide. Current gold standard for diagnosis of Alzheimer's disease relies on cerebrospinal fluid biomarkers that include amyloid peptide, total tau, and phosphorylated tau. This provide quick information about amyloid plaque and the neurofibrillary tangles associated with AD. However, these biomarkers are obtained through invasive process, which is called lumbar puncture, and this is not commonly available outside of highly sophisticated and specialized clinical setting. So on the other hand, PET combined with structural MRI, they are also widely used in clinical and research setting to diagnose AD. However, these biomarkers and neurofibrillary tangles provide high diagnostic accuracy of the presence or absence of the disease pathology, but these are influenced by various other pathological processes, including vascular dysfunction, inflammation, metabolic disturbances, and other factors that contribute to complex landscape. Therefore, not a single biomarker should be efficient for AD diagnosis, but multiple biomarkers are likely to be required to predict the underlying pathologies and disease stage risk stratification of clinical progression of the disease. Now, these complexities underscored the urgent need for developing non-invasive biomarkers for precise phenotyping of the disease to capture the full spectrum of the disease. Now, retina, the neurosensory layer tissue at the back of the eye, is directly connected to the brain through optic nerve and has been considered as a platform to study in central nervous system. Due to the common embryonic origin and the shared cellular, molecular, and functional characteristics with optic brain, the retina serves as a potential surrogate tissue for understanding the brain-related pathological processes. And advanced imaging technology, including UWFA, OCT, OCTA, retinal fundus imaging, that provides that non-invasive high-resolution retinal imaging that are capable of detecting subtle microvascular changes within the retina that are associated with neurodegeneration and cognitive decline. So given the lack of AD screening tools, that retinal images from the annual or biannual routine exams could be used to identify the main hallmarks of AD from the normal health screening program. So when we talk about that AD diagnosis or using artificial intelligence for better prediction of any disease or disease risk stratification, then we must consider the blessings of deep learning based methods. Now, what actually happened? These deep learning based methods evolved around 60 years back, and from that they have been developed along. So initially we had very 10 to 15 years back, we had very simple neural networks where we had simple nodes and simple architecture. But with the advancement of artificial intelligence, now more powerful computing network can be developed, and this could further increase the sophistication and complexity of the neural network. So specifically now we are able to take the single layer neural network architecture and essentially design much more sophisticated, deeper architecture with multiple different nodes and multiple different connections between the nodes. And by doing this, we could create more advanced and complex neural network architecture that could do really very interesting things and very very important things in healthcare, particularly for disease diagnosis, prediction, treatment response prediction, and disease prognosis. Now, the fundamental idea of the neural network architecture is here the individual node represents a small piece of information within the image. Therefore, considering a number of nodes, we actually could feed the total information within the image through the network through the architecture, and we essentially don't need to tell the network that which region is the most important region, which region is to be used for feature extraction. So the network has the ability to automatically learn based of the images and annotations or segmentations provided to the network, provided to the architecture. So this is really very powerful and efficient in learning the representation associated with the target. But the main problem of deep learning is that there is a real opacity of the network representation and what the network is learning, particularly in the domain when we are dealing with complex diagnosis, prediction, or prognosis problem in biomedical engineering. So there has always been a challenge to be able to understand how this structure looks, how this architecture looks, how the AI learning works, what the representation means, and how do we really take the trust the decision being made by this black box approach. Also, there is a major challenge to figure out where the network screws up and does not give the expected result and why it is not performing well. We don't understand the limitations of these algorithms quite simply because we don't have a sense that what is happening within the architecture, what the network representation actually mean. Now here I am showing an example where and also one thing that deep learning, we know that deep learning models are trained with a large number of dataset, but when the training samples are very small, then the network is not able to perform the expected output. So for example, here we used a model for using deep learning to predict anti-VEGF therapy treatment response in diabetic eye disease. And so we used UWFA images and we used OCT based images and we used and so we find that this is the class activation map. That is the region where the deep learning model gives more importance and has the more predictive power. So we find that for our objective was to identify the patients who were responders and non-responders of anti-VEGF therapy. But we found that the deep learning model, the here the red or the hotspots actually means the regions of more predictive power as identified by the deep learning model. So we found that these hotspots are shifting from one place to another, sometimes around optic disc, sometimes around the around vessels, sometimes the outer periphery of the vessels. And similarly for the OCT region, OCT scans, we found the deep learning model is providing more importance sometimes to the compartment, sometimes to the retinal tissue compartments. So that means the neural network model is uh, failed to identify the main region of interest which has the more predictive power. So, and here the sample size was very small. So what we are actually doing, that we are actually implementing the extreme power of deep learning, and we use the concept of deep phenotyping where we use the power of artificial intelligence for segmentation of different regions, for compartmentalization of different tissue compartments within OCT scans and vessel structures from fundus and UWFA images. Now, having done that, now we look at the features that are more interpretable and that have more inherent explainability. Now, our group is focused on finding novel interpretable ways in which we could find some visual attributes and features or patterns from retinal images that could provide information uh, above and beyond what the clinicians might be able to visualize in naked eyes. So, one of these interpretable features could be vessel tortuosity, which represents the vessel twistedness. It could be fractal features, which represents the heterogeneity within particular object, and it could be the texture information within some region. So here is an example where we used texture-based interpretable features to distinguish between super responders and suboptimal responders of anti-VEGF therapy in neovascular AMD patients. And we found that the texture of the super responders were highly heterogeneous as compared to the suboptimal responders. And this texture-based information that represents the heterogeneity within a compartment is very much helpful in identifying the super responders and suboptimal responders. So in the second panel, we can see for the baseline OCT images, within the texture of subretinal hyperreflective material compartment, there are lots of hotspots that represent the higher order of heterogeneity within that compartment, which has been reduced by a significant amount after 3 months of post-therapy. However, for suboptimal responders, the texture information remains similar. That means that is reflective of that this patient is not actually responding to the treatment. So having said this, that we are not ignoring the power of deep learning, but we are actually leveraging the power of deep learning, but using it to phenotyping, segmentation, compartmentalization of the image, but then extracting more interpretable handcrafted features from these images. Now, we have applied this handcrafted approach for a number of problems, including treatment response prediction to disease progression. So one of the works that I am going to present here that is related to Alzheimer's disease. And here our objective was to find association with retinal vascular features with Alzheimer's disease. And this work was done in collaboration with Emory Brain Health Study, Gazzetta ADRC, and Emory Cognitive Neurology Clinic. And we collected around 1,587 retinal images from around 1,000 participants. And out of them, there were 42 AD and 1,545 were control. So what we did, from the retinal fundus images, we extracted the vessel structure, and using deep phenotyping or deep learning based artery classification model, we classified the artery and the vein structure separately, and we did independent analysis of artery based and vein based features and find their association with Alzheimer's disease. Sorry. From the retinal images, we actually extracted different fractal features. Those fractal features actually characterize the architectural disorder or structural irregularity within an object, and more specifically vessel tortuosity, vessel twistedness, vessel angle, vessel connectivity, and vessel curvature features. And we identified a few number of features that were statistically significantly different between the two groups of patients. And using those features, we trained a random forest classifier model to identify to distinguish between Alzheimer's disease and control subjects, which acquired an AUPRC of 0.7 on the validation set. Now, our objective was to whether using oculomic features, whether we could improve the performance or we could provide better diagnosis. So we combined the different retinal fractal-based features or the oculomic features with different CSF measurements. And our objective was to investigate that whether this combination provides better performance as compared to the individual feature family alone. So we found that when we use CSF measurements only, we got a sensitivity of 83.33% and specificity of 65.78%, whereas using oculomic based fractal features, we got 100% sensitivity and 78.94% specificity. However, when we combine both the CSF measurements and oculomic features, we got an 100% sensitivity and 92.1% specificity. So this actually establishes the efficacy of oculomic features for Alzheimer's disease diagnosis. And so we identified that the fractal entropy is the most discriminating feature. Now, this fractal entropy actually characterizes the structural irregularity or the architectural disorder of an object. So as the feature map shows that for AD patients, the fractal entropy is highly expressed, reflected by the warmer color tones, and we see that the prevalence of these warmer color tones are high for the AD case. That means the vascular features or the vessel structure are more twisted, convoluted, and have complex architecture for the AD. However, for the control cases, the feature expression is more lower, that is reflected by cooler color tones, and it represents that the vessel structure is more regular. And we also performed a correlation analysis between these fractal features and different CSF measurements, and we found that the fractal entropy is correlated with the CSF measurement total tau with a Pearson correlation coefficient of 0.59. So this increased fractal entropy is might be due to the total tau effect that may progress to the development of cerebral amyloid pathologies or structural disorder in capillaries and in vessels, and that might be the reason for dysregulation of the vessel architecture that has caused the increased entropy. Now, having said that, we just wanted to check that whether how these AD and ADID subjects are influenced by different external exposures and different environmental factors. So emerging evidence highlights the critical role of external exposures in the environment, including air pollution in the pathophysiology of AD and ADID, and documented effects of air pollution include the activation pathways related to oxidative stress, inflammation, and epigenetic changes, which contribute to ultimate neurodegeneration or cognitive decline. However, our understanding of how many external exposures interact with the PM 2.5 and casually affect ADRD is still limited. So here, this is an image from Google that where we show that the different environmental different external exposomes have significant proportion of ADRD mortality variance in the US. From the bivariate heat mapping, we see that the PM 2.5, that particular environmental exposure interacts with the social vulnerability index to determine the ADRD mortality. Now, so in this experiment, we actually developed an oculomics pipeline to extract different retinal vascular features and to find out their interaction or their association with PM 2.5 exposure in EucoBiobank data. So, around 6,400 participants. So, we found that the PM 2.5 was linked to different retinal artery-based predictors of risk with no vein predictors were identified. So what we actually did, from the EucoBiobank data, we collected different retinal images, and from these retinal images, we extracted different vessel density, caliber, fractal-based different features, and we used completely unsupervised hierarchical clustering approach. So as you observe that the hierarchical clustering clearly shows two separate clusters, and we found that this red cluster that actually clusters all the subjects that are more exposed to that are mainly urban based population. That means those subjects are more exposed to PM 2.5, they are more distant from the sea coast and more educated. However, for the green cluster, that is that actually consists of more rural population where the PM 2.5 is lower. That means environmental exposure, air pollution is lower, where the distance from the more greenery is there, and there is more distance from the sea coast. And so, as I mentioned, that we identified a number of artery and vessel features that were linked to PM 2.5. And we also identified that not only PM 2.5, but some other exposures that like PM 10, nitrogen dioxide, those features are also linked with Alzheimer's disease, and that shows statistically significant difference between the AD and control group. And we also did a time to event analysis where we found that we could identify two clusters. One is as I mentioned, that the cluster that are more exposed to that are more consisting of rural population, and another was the urban population. So we found that for rural population, the risk of developing AD and ADID is less as compared to the urban population, which are higher risk of getting AD and ADID. So next our objective was to whether we could predict the chronological age from from this model, and whether the biological age acceleration could be used for predictive marker. Now, the biological age actually reflects the cumulative molecular and cellular deterioration underlying the functional decline, so distinct from the chronological age. And so what we use the emery cohort as a discovery set, and we use the AREDS and BRST cohorts for model training, and we tested validated the model on the Yuko data. So what we observe, so here we actually calculated the delta age. So delta age is the difference between the biological age and the chronological age, and uh, with the delta age we actually calculated the we actually from the delta age we classified the subjects into three groups. One is the resilient groups, that is less than 20 percentile, normal group 20 to 80 percentile, and accelerated group, that is greater than 80 percentile. So, and we developed a risk prediction model. So, to find out what is the survival probability of the high and low risk, and we found that the high delta age, that means the accelerated age group, has the lowest survival, or the have greater risk of getting AD and ADRD progression, whereas the resilient and the normal group have lower risk of getting AD and ADRD progression. And we also did a proteomic analysis to compare the accelerated age group versus others. So we did differential gene expression and pathway analysis using O-link O-link serum proteomic levels, and we identified that the telomere length is there is a statistical significant difference between the accelerated age group and the others group with respect to the telomere length. And we through the proteomic analysis we also find out a number of uh, genes that were differentially expressed between the two groups, and that show statistical significant difference between the two groups of patients. And also on the pathway regulation analysis, we found that a number of pathways, including the cellular response to stress, cellular response to different chemical stimuli, and other pathways are actually down regulated for the AD patients. And this down regulation, actually these pathways are uh, uh, connected to the uh, connected to tau pathology and neural the different neurodegeneration. So the down regulation of these pathologies actually represent that uh, for these patients, that means for the high risk patients, there is a higher order of getting this cognitive or neurodegenerative condition. So there are lots of limitations of this study. And so the main takeaway is that we could use oculomics, that is an emerging field where we consider the retina as a potential window for investigating different neurodegeneration and cognitive decline related changes. And we could do it in an non-invasive way, and we found strong correlation between different artery-based features with references of measurements, which also established that these features have potential in identifying the AD patients, and also diagnosing that which patients are at the high risk of AD. And also so we also have explored that the different environmental exposures influence actually the accelerated aging, which is correlated with higher AD risk. So there are a number of limitations of the study. So one of these, that the oculomics model that we developed, that for 80 diagnosis, that had limited number of 80 patients. So and it is a single institution data. So we need to validate the model on multi-institutional data, and we need to further investigate that on longitudinal scans, that uh, what are the structural changes within the vessel architecture that is going on as the disease progresses. And these biomarkers also need to be validated across diverse population. But overall, I think that I could provide some knowledge about how artificial intelligence could be applied for disease diagnosis, particularly in Alzheimer's disease space, and how accelerated aging is, how from the oculomic based model we could identify the person who has higher chronological age, and how this is correlated with higher AD risk and enhanced dementia prediction, that can serve as future biomarkers for neurodegeneration. So thanks. And I'm happy to take any questions.
Thank you so much for that great presentation, Dr. Sil Kar. I want to remind folks that you can post your question in the Q&A box. I don't know if you can still hear me. I think are you here? Okay, perfect. We lost you for a second. One of the questions that's also in the Q&A that I wanted to ask you too is, when we think about retinal imaging, how representative are the existing data sets? A lot of times when we look at correlations between one data source and prediction for cognitive decline, we're always questioning, the generalizability, whether there's bias in the data. And similarly, there is a question, how can we help expand your research into a lot of different communities, including Asian American communities?
Yeah, this is a nice question. So regarding the generalizability, so this is, I have mentioned that what we are in our previous work, in one of our previous work, we have identified the stable features that around different institutions. So we collected features that were not discriminating, but also that were more generalizable across the different data sets, and we are considering only those features that those so actually what happens, these features are affected by the scanner variation and different parameters of the scanners. So we only consider those features where the feature expression remains same for different scanners. So that ensures not only discriminability but also the generalizability of the model or the feature stability. Yeah, and the another question, I think, what was that? I can see in the Q&A section now that Asian
Yeah, the question was
right?
about expanding your
Yeah.
research into Asian American communities.
Yeah. So we are currently working on subset of these subjects and developing the population specific model, say for this we are building, so to find out that what are the structural differences, whether there exist between those population. And also we have a very large cohort, that is PCARS cohort, that is particularly in India and based on South Asian population. So we are also collecting their retinal images, and we are trying to find out their association with Alzheimer's disease and other systemic diseases also.
Thank you. There is a question about data splitting. I think it was brought up when you were talking about the proteomic slides, whether you did any data splitting.
Yeah. So it is we did the 75-25, that is data splitting, and we developed the model with the 70% of the data, and rest were used for internal testing, and also we used the UCO for external validation.
Great. And then I
So
think,
are all this retinal vessel in the studio?
oh, please go ahead. Yeah, there was a question
So
about
yeah,
whether retinal
this
vessel
is also a
images are
good
2D.
question. Yeah. So all 2D images. Okay. So for that we extracted the 2D features mainly. But I mentioned that for OCT we are also, for as part of the project, we are also prospectively we will collect different OCT images, and those OCT images are volumetric measurements, and from OCT images, from those 3D volumes of OCT scans, then we will extract different 3D features, and those features will provide. Okay. So there is another question I see. You might have already covered the email research to collect data. And for how many years? Okay. So here the question is that whether how we collect data from subjects where they have corrective glass, and for how many years. So this study that Emily Brain has studied that started before COVID around 2018, and the data were collected. This is totally retrospective cohort, and the data were collected up to 2020. And now, and after COVID, there was some gap in the data collection, and we will start the data collection prospectively also. So, but the retrospective cohort that we collected, there we excluded those patients that have any ophthalmic condition like glaucoma, age related and there are different retinal problems, macular degeneration. So we have excluded those patients so that we could identify, and the only included the patients who did not have any type of retinal disorders. Yeah, okay. So another question is, how did you handle the data imbalance? Oh, this is also a nice question. So as I mentioned, that the number of 80 participants is limited. So we used SMOT, that is, yeah, this is a machine learning approach where we artificially generate minority classes and to make the dataset imbalance. So that is moot, and that imbalance, data imbalance handling technique was used here.
I wanted to add a question. This is obviously a very promising uh, line of research, especially if we think about a new type of biomarker to assess cognitive decline. What would be some of the unresolved scientific questions that you think this field needs to explore in terms of using retinal imaging or other types of related biomarkers in order to understand and predict cognitive decline?
Yeah, that is a very nice question. So that is also one of our study that we are working on. So I have talked about AD patients. So these AD patients are symptomatically AD. That means they have clinically AD symptoms, and also they have cognitive impairment. But within these AD patients, there is one subgroup that is called asymptomatic AD. That means they are using the CSF AD biomarkers, they have that cognitive, they have uh, diagnosed as AD, but they don't show any kind of cognitive decline. So this subgroup of patients, that how could we identify this subgroup of patients, and how whether this subgroup of patients are more AT-like or more control-like. So this is very important to identifying this subgroup of patients and finding that what are the what is going on between, what are the differences in with respect to retinal images and with respect to the brain developmental changes that distinguishes between the symptomatic AD and the asymptomatic AD, which don't have any cognitive decline, any cognitive impaired, but have biomarker positive AD. So this is a very important field that we need to work on in future.
Great. Let's see if there's any other questions. Just to remind folks, you can use the Q&A button on Zoom to type your question. And if not, I want to thank you again, Dr. Sil Kar, so much for this great presentation and giving us opportunity to learn more about your work. I want to also remind folks that this is being recorded and will be posted on our YouTube channel for later viewing in the next few days. Our next webinar will be Thursday, December 11th, with Brianna Vecchione, who is a technical researcher with the AI on the Ground team at the Data and Society Research Institute, with her Talking to Machines, How LLMs Are Changing Mental Health Support in the Age of AI. Again, Dr. Sil Kar, thank you so much, and thank you all for attending today.
Thanks. Thanks. Nice presenting here, and thanks for inviting me.